Holes.
Yeah, is that the new show? Change the title. Change the show title right now.
Hey, we're live, everybody. It is time for us to change the title of the show. It is now going to be called Hairy Face Holes.
Hairy Face Holes.
Oh, wouldn't you like to know what the conversation was before I hit the start button? We are here and we are ready to start this episode in... Oh, let me make sure that I've got my music ready to go. Of course I don't. Of course I don't. Hold on one second. There we go. Okay, that'll do it. Later. And we can start this show in... Oh, come on. Cancel Apple. Apple's being a pain in my booty right now. Apple. Wow, it really wants me to sign into the iTunes store.
No.
Really?
Oh, no.
I don't know what I wanted to be doing right now. How about we just quit? Cancel. I don't remember my password to the Apple store. That's why I never sign in. I just want to use iTunes. Why are you being so rude?
It's not going to let you.
It's not going to let me. This is some compelling video, everyone. Oh, this is what I came to do. Click this button. Oh. It's time for bed, young child. We're going to be starting this interview as soon as I can sign into the iTunes store. Maybe I'll have to play it a different way. Maybe I'll play it a different way. Hold on. Oh my God, and my cat is way all over the place. Un momento. I'm going to Macintosh hard drive. Music. Music on my computer. Here we go.
Go to Classroom, please. Go to Classroom, please.
All right. I can play this. I can do this, I think. We can do this. Okay, everyone. We can make this happen. In 3, 2... This is TWIS. This Week in Science, episode number 723, recorded on Wednesday, May 29th, 2019. The Science of Sex. Hey there, I'm Dr. Kiki, and tonight on This Week in Science, we are going to fill your head with helpful dirt, fish skins, and sex. But first...
Disclaimer. Sex! It's that thing your parents did that made it possible for you to not want to think about your parents having sex. But they did it, and did it in such a way that made you possible. And that is not the only reason you exist, but that's the beginning of the story. Turns out your grandparents also had sex. If not for them having sex, your parents would not have been able to swap chromosomal chemistry in the first place, which means that at some point, your great-grandparents, all 8 of them, needed to get busy in just the right way to set in motion the sexual behaviors of offspring that, again, no matter how you choose to or choose not to think about it, is how you got here. Sex is the reason just about every person you could ever meet is meetable in the first place. And with all of this, sex is the coming together of more than just 2 individuals. It is a merging of genetic information of tens of thousands of generations of hominins, with inflexing and outmoding of genes, with specialization, mutation, and epigenetic adaptation. To put it most bluntly, the simple act of parents having sex is the thing upon which most of life, love, and the evolution of life on this planet is based. That and This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I wanna learn everything. I wanna fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I wanna know what's happening. What's happening? science. What's happening? What's happening? What's happening this week in science?
Well, good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again to talk about the wonders of the world that are filled with scientific curiosity and information, all the things that we have discovered, are still discovering, the questions we're asking. And on this week's show, once again, there's a great show up ahead. I have stories tonight on what makes us human and what might make us happy. We also have an interview this evening on sex chromosome science. It's going to be very thrilling, very sexy. What do you have for us, Justin?
Oh, that's a great question. I have a coral acid trip. I have some dirty fat news and why Neanderthals went away.
Let's be
Yeah.
dropping some coral this evening. And Blair, what is in the animal corner?
I have horngry bats. Explain what that means later. I have fish skin, and I have turtly hungry chimpanzees.
We always love a good chimpanzee story.
Well, you
I
won't
think-
anymore after tonight, but I'll find out why.
Oh no.
All right. I love it. I am waiting. I'm waiting for that one. But before we jump into the stories and all this fun stuff, I would love to remind you that if you are not yet subscribed to the This Week in Science podcast, you can find us on iTunes, on the Google Podcast portal, the Apple Podcast portal, Stitcher, Spreaker, Spotify, Pandora, Radio.com, TuneIn, all the places that podcasts are found. You can also look for us on YouTube and Facebook, or just visit TWIS.org. And now, I would love to introduce our guest for the evening. We are joined by Dr. Melissa Wilson, and she is an associate faculty in the Biodesign Center for Mechanisms of Evolution, an assistant professor at the School of Life Sciences at Arizona State University. Her research interests include sex-based biology. She studies the evolution of sex chromosomes, the X and the Y chromosomes in mammals, why mutation rates differ between males and females, and how changes in population history affect the sex chromosomes differently than non-sex chromosomes. Without any further ado, Dr. Wilson, welcome to the show.
Thank you so much for having me.
Yeah, it's wonderful to get to speak with you. We talk a lot about the biology of sex. Blair brings lots of stories to the animal corner about sex in the animal kingdom. We talk about human evolution. We're always discussing this topic, but it's really good to get someone who is an expert. How did you come to study sex-based biology?
Oh gosh, it was kind of a twisting path. So I did my undergraduate in mathematics, actually. Small one. Go. Sorry. I mean, what you were talking about at the beginning, yeah, I do have a child, and someday she'll have to think about how she got here. And I'll revel in that when that happens. So I majored in mathematics as an undergraduate, and I did a summer research experience where I learned about some biology, and I ended up applying to a bioinformatics, so a biology and computer science program, and they let me do rotations. And so I did a rotation learning about yeast genetics, and it smelled like baking bread, and it was great. And then I did a rotation learning about Arabidopsis, which is a mustard weed. And then I did a rotation with Dr. Katerina Makova studying sex chromosomes, and like it was over, right? I knew that I would be studying that for the rest of my life, and I have been, and there's not a point where I don't think about that or relate to things. To get back to your sexy, you can't have sexy without X and Y. So it's kind of where I'm going.
Bringing it all together. So getting interested in the mathematical informatics side of biology. I think coming from a biologist perspective, like everything in my life was always wet and squishy. How how is it to study biology from this other perspective of we're looking at the informational side?
Yeah, I think that's the thing. When people come into the lab and they ask to take a picture of me doing my lab work, right? They want me to put on the lab coat and go into the wet lab and show them some samples. And I'm like, no, I do this. I work on the keyboard and we sit on the computer. And I think that can right now, please go. No. I appreciate you all. Thank you.
We're all, except for Blair, we're all parents here.
Yeah, I work with kids, though.
Yeah.
I know what it's about.
It's a very different kind of thinking about how science is done, but it's where science is going in the future, right? All of the genomic testing, when we look at genetic ancestry, when we look at risk factors, when we try to think about causes of disease, right? We're looking really not anymore at a single gene, right? We're looking across, if we do the whole genome, 6 billion sites, and then we look at multiple individuals. So we're looking at maybe 10,000 individuals, 100,000 individuals across 6 billion sites. And you can't do that by hand. So you have to do it on a computer. And so we write programs and we look at things. And if you come to my lab, I'll concatenate a file to the screen and it'll go, and it'll look like the Matrix and I'll feel really cool and you'll be really impressed. But I would never really do that on my own, other than to just feel like maybe I'm not so big of a nerd all the
time. But being a nerd this is important. We like being nerds here.
So on that note, in doing sequencing, one of my favorite things is Illumina on their Nextera sequencers. The machine makes a really happy sound when you start a run. It's like, da-da! And like, so you get like this moment of like, something's happening. It's really exciting.
It's like an Instapot for genomics, right?
Totally.
From an evolutionary standpoint, your perspective also, which is better, sexual or asexual reproduction?
Oh gosh, not it. In fact, in next week, I'm going to a whole meeting by the American Genetic Association on sexual and asexual reproduction, because we still don't have great ideas about, so we have lots of great ideas, but we don't have answers to why has sex evolved so many times, and why does asexual reproduction persist, and why do some populations switch between sexual and asexual? And if you get stuck in sexual, are you really stuck? Can you get out of it? We see whip-tailed lizards with completely female populations, so they got out of it. You know, what do we do? I don't know.
And then you have animals that jump in and out, like the anaconda at the New England Aquarium this past week, who went through parthenogenesis and had a virgin birth.
Yeah.
Or just yeast, which you were studying, right? Well, yeast can flip back and forth.
Yeah, no, I think it's fascinating.
Yeah, it's such an interesting question, because when you think about all of the life on this planet, there are so many different strategies, and but I think it's a fascinating one. You know, evolutionarily, fitness and survival and more generations is the best thing. Which is better?
It does seem though that there should be another goal, which is just like, okay, this was it, but now I'm just gonna live forever. Like that's the one. That's the goal. Like I'm done with this reproduction thing. We're just going to ensure that we live forever this generation.
Well, jellyfish have kind of figured
jellyfish.
That out.
Yeah.
We started talking online about
hydras.
Yeah, right. Those interesting ocean creatures. But we started talking online because I came across interesting facts about platypus reproduction, and their genome not only is as interesting as their exterior, but they also have 10 sex chromosomes.
Right.
Why?
So X and Y, and then what?
X and then X and Y, and X and Y, and X and
Wait, what?
So X and Y is a term we use that refers to any species where the males, so the sperm producers, can make a gamete, a sperm, with either one kind of chromosome complement or the other, so an X or a Y, but it could be two X's and two Y's, or in the case of the platypus, 5 X's and 5 Y's. And the platypus gets all of this press, right? Because everyone knows about the platypus and it's exciting and it's an egg-laying mammal and it's adorable. But there are other egg-laying mammals, the echidnas. And so what we think happened is that platypus and echidna in the common ancestor, they got one X and Y chromosome pair, and then they added a second pair, and then they added a third pair, and then platypus diverged from the echidna, and it added a fourth pair and a fifth pair. But not to be outdone, the echidna added a fourth pair independently. It added a fifth pair, and it lost one of the Ys.
No. So there's 9.
So female echidnas have 10 X chromosomes, and male echidnas only have 9. It's their only claim to fame. What else? I mean, the platypus gets all the attention, and the poor echidna.
The echidna's amazing, though. They have this little straw
The
face.
beak, they're like straw beak.
Gosh.
And they also lay eggs, and they're pretty awesome. But not that the platypus is not awesome, but we don't now. About 20% of the whole platypus genome is wound up in a sex chromosome, in one of those 10 sex chromosomes, as opposed to us, where it's 5%. So it's pretty exciting.
So those chromosomes are very important to them. For us, I mean, when you're looking at the sex chromosomes versus the non-sex chromosomes and trying to determine their importance for a species or for evolution of a species, how do you start to figure that out?
So we have some things that we used to think were true. We used to think that if you became a sex chromosome, eventually you would, one of the 2 sex chromosomes would decay over time. Like our Y chromosome is much smaller than our X. The human X chromosome has about 1,100 genes, and the Y has about 27 unique genes on it. This is what being a science parent is like. You have like small children and small animals and large animals, and you figure out time management sometimes. But one of the things that I think is maybe most dangerous to people who don't study evolution all the time or don't think about it is to say, what was the purpose of this? So adaptation is a very strong force, right? But many, many things happen because they didn't hurt the species enough, right? So it's not only evolution of the good enough, right? That's probably what it should be, not evolution of the fittest. It's like evolution of the good enough. But also those that just, we in populations with a small enough size, if you were good enough to get some, you would have kids, right? Like that's it. And it doesn't matter if maybe your cardiac output wasn't the best. And it doesn't matter if you weren't the most beautiful, or we carry a lot of pieces in our genome that are there for no good reason.
Well, especially if it's something that shows up after you've reproduced, right? So there's all sorts of things that show up after you've already given your genes to the next generation. So it's kind of moot.
Yeah, then they can be even harmful, right? So it's not only that there's neutral things floating around, so lots of neutral things, there's lots of deleterious things that stuck around, and things that are trade-offs, right? Something that maybe was really good and helped you have a ton of reproductive success when you were younger, but that same thing could also be harmful if suddenly now you're living to be 70 or 80 or 90 years old. So that was not a great answer to your question, but that I sometimes don't think about exactly. I think about what's important on the things that have been retained on our very gene poor Y chromosome, and are those things critical? And it turns out some of them really are. There's a few of those genes, X and Y used to be identical, and there's a handful of those genes, roughly let's say roughly 17 of them that have a copy on the X and a copy on the Y. And if you don't have a functioning copy of the X and the Y linked copy, like you don't function as a human. Like the Y lost pretty much everything it could other than this small set of things that we really need, except for one gene, which as a PhD student doing some research, we looked at how all of the genes are conserved on the Y chromosome. And there was one gene, AMELY, amelogenin Y, that has an X-linked copy, amelogenin X. And the X-linked copy is involved in helping make the enamel on your teeth. And if your X-linked copy is broken, then you can get broken enamel on your deciduous teeth. But if the Y-linked copy is broken, nothing happens at all. And in fact, some human populations have totally lost the Y-linked copy. It's deleted. It's completely deleted in some human populations. Why, you might wonder, might I care so much about this? I care so much about it because it's one of the few genes that I think we might lose on the Y chromosome. And it is, or at least was for almost all of our forensics history, the marker that people used to try to sex the sample. They would say, what? Oh no! What? Why did they not talk to an evolutionary biologist? We could have saved the day. But no, the one gene that they chose on the Y chromosome as the marker for the forensics kits that we use to say, if you have this marker, you probably came from an XY individual, and if you don't, you came from an XX individual, is the one gene that evolutionary biology tells us is going to be lost or already is lost from the Y chromosome. You're very welcome.
Missing. It's already missing from some human populations.
Yeah, I hope you don't have serial killers listening to this.
Probably do. Probably do.
Why is it though? I mean, we've heard there have been stories through the years that the Y chromosome is disappearing, and you would look at the number of genes that it has compared to the X chromosome, and it seems like it. But then you see these very well conserved linked genes that seem absolutely important. So then maybe it's not going to disappear. I mean, what's happening to the Y chromosome? Why is it shrinking?
So we think we know the why it's shrinking, which is that every other chromosome 1, you get a copy from mom and a copy from dad, and they can swap DNA to fix errors or mutations. Chromosome 2, chromosome 3. But chromosome Y and X, they can only swap at their tips, and they can't swap on the rest of the chromosome. So if there's a mistake on the Y, you can't fix it. In XX individuals, they still have 2 X chromosomes, so they can swap, so the X is fine. But the Y, when it accumulates mutations or deletions or even insertions, it can't get rid of them. And so we think that that's potentially why. But when it comes down to it, if we lost the Y chromosome, it wouldn't even be a problem. The Y chromosome is not males. What we care about is whether individuals are developing testes and making sperm. And that can happen if you move SRY to an autosome. It can happen if you move a chunk of genes. There's been species, there's a spiny rat in Okinawa that has lost the Y chromosome completely. Males are X not. They just have a single X chromosome. And they're still fine. The populations are totally fine. So when it comes down to it, we're not terribly concerned whether we lose. The Y is just a home for genes. And that home, those genes can find a new home.
What?
And we can have neo-sex chromosomes. We get brand new sex chromosomes evolving now. So, yeah.
When you look at, I mean, there's, in evolution, we have these diversity of alleles and diversity of, designs. Like with the platypus, they have 10 chromosomes, and you've talked about like the echidna now getting another one and then losing one, and it's a very dynamic system. And I mean, how it's really stunning to me when we think of our chromosomes as something that makes us human and makes us who we are. The possibility that they could change, and that we would still be human. We would still be who we are. It's fascinating.
Yeah, I think we get stuck in this idea that it's genetic determinism, that your genes determine who you are and that your chromosomes determine who you are. And that's really not true at all. If we think even about sex chromosome copy number variations across humans, they're not uncommon. So individuals who develop ovaries and are assigned female at birth but have a single X chromosome are as common as 1 in 2,500 humans on the planet, right? And
really
that's
common.
Turner syndrome. And individuals who are assigned male at birth develop testis and have 2 X chromosomes and a Y is Klinefelter. And that's as common as 1 in 500 humans. So these things are not uncommon. And that's just the gross, when I mean gross, like large, not gross because they're super cool, chromosomal variations. And then we can have individual genes that are moving around. We can have genes that are turned on more than typical, genes that are turned or reduced expression. So I really try to avoid saying something is normal or something like this. This makes us human because there's just so much variation. I love that you used alleles also. That's probably one of the things I love most, right? A variant of a gene. We have so many variants. And so we did a back of the envelope calculation once about if you took every single site, 6 billion sites in the human genome, and you took the average mutation rate and the number of people alive on the planet, every single site in the human genome would be variable in more than 100 people across the whole planet. If you could sequence everyone on the planet, every single site in the whole human genome would be variable in more than 100 people. So what's normal then?
Yeah,
That's a lot of diversity right there.
But what you're talking about too is this is the thing that we really hope the genetic code would be, is an absolute behind the scenes blueprint from which we could solve every mystery, cure every disease, and predict everything that would come from any individual's code that they had. And what we've learned is, this is the plasticity is there, as well as the sort of cascade effect of genes working in tandem with each other to have different affects.
Yeah, 100%. Yeah, I mean, human geneticists, we lied. Yeah, definitely lied through our teeth. Okay, we didn't, right?
No.
Certainly, we thought we were going to figure it out. We did. And when I started, so the number of genes in the human genome has changed dramatically throughout my experience in genetics. And it's still changing. And I just hope that we start to do a better job of appreciating all that variation and exactly how things are interacting, so that we actually can. We have made tremendous strides in treating a lot of human diseases. We have. And I believe that if we start to take a step back from the single gene, single phenotype, or single syndrome model, we'll start to solve or at least find treatments for more of these. It's not just a puzzle anymore, right? It's like a multi-dimensional.
And there's an interesting correlation I think of when I think of like our understanding of nutrition and health and what we now understand about the microbiome. Mm-hmm. And within genetics, there's a microbiome of mRNA and RNAs and motor proteins and all sorts of other smaller actors that are causing upstream, downstream, whichever direction you look, effects that hadn't occurred to us are the motivators for how things actually function. So we are, as we are just scratching the surface of nutrition when it comes to the microbiome, we're just actually scratching the surface of genomics, because there's a whole bigger, more complicated layer beneath just the genes themselves.
Yeah, and I think so one of the things that, because we study sex differences broadly, one of the first questions I often get is about how do you know that it's sex chromosomes versus hormones, right? So we know, especially, there's large differences in testosterones and androgens and estrogens and progesterones. And so aren't the two just completely linked together? If you're XX, then you're going to have this set of hormones. And if you're XY, you're going to have this. And it turns out that's really not true at all. And we see differences in gene expression on the X and the Y during development before you even have gonads developing. So now we have this added layer where we have X and Y have their own unique gene expression. Then those are modulated just like everything else in the genome by androgens and estrogens. And then there's this crosstalk. So it's kind of this wonderful symphony. And we just have to pick apart, you know, what's the violin contributing and what's the cello contributing and where are the timpani drums, right? Like where are all of these things? And that's what we're trying to do now. Like to me, that's, I don't think it's that we're not finding the things. We just have to listen and try to tease apart those different aspects.
Do you think with the amount of variation that there is inherent in the human genome that you know, that we will eventually get beyond the single gene, single variant kind of system? Do you think with bioinformatics, are we going to be able to eventually really address like the multi-layered, multifactorial aspect of the problem?
100%. I think that's where we're going now. That's where the leading edge of bioinformatics and genomics is going, is trying to figure out how do we look for networks of interacting genes, and not even just interacting genes, but interacting isoforms. So interacting variants of the genes. So not even just alleles. So for a gene, right, you can have different alleles, but then you could have different sets of, well, exons put together. Sorry, I'm getting too complicated, but right. So how do we look at the variants of the gene? How do we look at the particular mutations in a gene? How do we look at how those are interacting with our hormones, with our environment, with our nutrition, with stress? It's not going to be solved in our lifetime.
Okay.
Or ever. I'm going to say that. You know what? Come back to me when I die and tell me if I'm wrong. But I think we're making headway on it, and we're trying to figure out.
Wait, won't that be too late? I mean, it'd be a really one-sided conversation at that point.
Well, maybe we will have perfected the brain in a jar stuff that we were talking about a few weeks ago, and then it'll be fine. We'll just type it in on a computer.
About the definition of death. But
what are we? But she was going to say, but we're trying to figure out. And you interrupt. I was like, you're leaving me hanging.
No, we're really trying to. Some of the things we're working on right now is to look at not only the mutation being associated with a disease phenotype, for example. One of the things we're looking at is liver cancer and sex differences in liver cancer, but we're looking at the interaction of that gene, that allele with other alleles, and then every possible combination of alleles. So those sets of combinations get astronomical really fast, and it used to just be impossible to do that kind of computing, but we're starting to get to be there. For example, at a meeting a few years ago, there was a large project called the 1,000 Genomes Project, which eventually ended up sequencing more than 2,000 people, and it took multiple labs, many years, and tons of compute time. And Google, because they could, decided to redo the whole project and like tell us how fast and wonderful they did it in like a month, right? They just redid this decade-long project, and the entire presentation, right? I'm gonna get in trouble, but the entire presentation, I'm watching this, and they didn't show a single new thing. They were like, look, we redid this figure from this original paper, and then we made this figure, and then we made this figure, and then we did this. And you know what? I did it, in my spare computing time because I'm Google and I can. And and on one hand, I had been waiting for and the next thing we did was this. And they may be working on that next thing, but the fact that they were able to replicate a decade worth of work in a month was just mind-blowing, right? And so we're going to be able to, if we don't kill the planet or ourselves first.
We're gonna work on that.
Yeah, minor details. Yeah.
The current next generation of sequencers can do the Human Genome Project or the 1,000 Genome Project over a weekend.
Science fair project.
The nanopore stuff alone is transforming the amount of data that we can generate. Okay, so now we've generated mountains of data. What do we do with it? there's mountains of data. Oh my gosh, there's so much data that needs to be then sorted through. And we have humans who are trying to employ more and more machine learning. But at this point, we are way behind the amount of data we can actually generate from this to even understand. So I'm thinking, just pause it, hold up, let's just generate a ton of data. A generation from now, people will be able to mine that like the Google did and do it in a month. Let's not try now. Let's just generate the data now and let somebody else worry about the apartment complexing it and figuring it out and making it work later.
So yes, but no. I mean, one is where do we store it, right?
Yes.
Who's going to pay for it? So some of these big projects that are publicly available data, I haven't been able to get access to because it's just such a large amount of data. It would be like saying, I would like to fit a blue whale in my living room. Yeah, sure, I'd like to do it, but one, how on earth am I going to get it here? And once I get it here, how many small pieces I'm going to chop it into till it fits in here? Is it even going to make sense? How am I going to put it back together? And good Lord, it's going to stink, right? Like, it's just going to be awful for everybody. And that's almost what it's like dealing with these massive data. Right. But we could generate it, right? Well, that's one thing is we could generate it, but the chemistry and the technology is continuing to get better. So so should we generate it? Should we generate the data if we don't have the computational approaches to analyze it right now?
And so I guess a part of what you're saying too is it's like we ran into this while we were talking to the folks that were studying, I think, black holes or dark matter or whatever. They're generating a tremendous amount of data that is going to be 10 years of analysis after which they've all left the project and moved on to something else. And like they realize we created more data than we can digest. Okay. So yeah, one of the ideas I've at least been hearing about is the idea that you don't have to have the whale in the living room, that the whale exists in one place and everybody goes to this sort of cloud-based, you need like a public research cloud that's just ginormous to store all this stuff that people can look at these things there and never have to have anything local or anything moved. Because
so the NIH, the NIH is working on that as well as other institutes. There's the NIH Strides initiative where people who are working on the data storage and the data analysis side. But some of the challenges still are the cost, right? Yeah. So it's there, but someone has to pay for it. And who's going to pay for it? And even let's say I just make a copy of one of the smaller data sets right now. It's going to cost me $10,000 a month. And I computed Amazon costs, what it would cost my lab to do the kind of work we're doing if I paid Amazon to do it. And it'd be about $15,000 a month. Yeah. So it's not free. Yeah. And it's one of the things I'm actually most concerned about with my lab going forward is the data are getting so large. Where is it going to be? Who's going to be able to access it? Is it just going to be big labs? And then what does that mean for the future of science? I think we're coming together to try to find solutions, but that's a concern right now for pretty much everyone doing genomics.
But isn't it also amazing that with all the advances in technology and computing power, that's the bottleneck?
Yeah.
Like, that's kind of cool that the bottleneck is actually information storage and handling now. I mean, that's an incredible place to have pushed the bottleneck to in our ability to do the deep dive.
Yeah.
I don't know if it's ironic, but it's maybe irony adjacent. Definitely.
Yeah. I would say another bottleneck though is training students. So the students in computation, computer science fields, don't have enough of the biology background to know what the assumptions are. Yeah. And students in biology are still not traditionally trained in computing. And so it's one of the things we're working on that it's been kind of my pet project, and I'm going to push forward over the next few years is how do we as a community, and there's a lot of people that are working on it, right? I could list a ton of people that are working on it, but how can we integrate better so that it's not black boxes? Because they can't ever be black boxes. I mean, they will be to some people, but how do we get students, and especially students that have been underrepresented in science who haven't been able to ask their questions, right? That's the one other thing, if I could jab it in there, is that in sex differences research, most of the questions have been asked by only one sex, and that
Mhm.
limits the questions that are asked.
Mhm.
Almost all the clinical research that's been done has been done in European males, and that limits the kinds of things we can ask and the questions that we can ask, and also limits the utility of the results. And so one of the things we're really trying to do, ASU, I drank the ASU Kool-Aid on it's who we include, not who we exclude, that we pride ourselves on, right? And to do that, we actually have to develop this training, because the questions I think about and that I'm interested in are very different than the people who are in my field who I admire but are 30 years my senior, and they're very different than the people that I'm training. And so that's the thing that I would like to see us as a community work forward, to have more open source free trainings in computational life sciences. And that's a nice thing about the genomics field. Like we have a couple sticklers, but we're pretty good at, okay, yeah, we have some real a-holes. We do. Everyone
There's work
does.
to be done everywhere.
Yeah, there's work to be done,
You
but
know.
we're working on it, right? Like, and there's enough of us that are like, you know what? I would rather have I would rather science be a great place than not. And I'm willing to put in the time and effort and give up some things because of that. And so I'm hoping that those voices will continue.
I wonder how we've talked with people in the DIY bio, that kind of biohacking community where people are starting wet labs in garages, or creating office spaces that are communal research spaces, and that's happening for biology in general. For people are starting to use CRISPR in their garages and in their kitchens. But I wonder if this open training for computational biosciences could also be implemented some way in this, DIY fashion.
I would love to see that. And I mean, there are some really great resources already. So it's not that some of the resources don't exist, but how do we convince middle school and high school and college students that that you will be able to do cool science using your computer and not just in the wet lab, right? That's a little bit of it, but I think we are getting there. I do. I think, yeah, DIY bio is I could CRISPR my dog. Like, oh, okay, I'm not going to, but right?
But you could, yeah.
How we can generate all the data in the world, like we can now. It's amazing, and we can do this cool science, and we just want to make sure everybody can.
Yeah, I think
right
there's a lot of potential out there, considering that computer literacy is the baseline of just life now. And I think that there was a lot of barriers, especially to women participating in computer science in the past. Felt like a male dominated field, not for women. I don't feel like that's so much the case anymore, not nearly as much. We've talked about it on the show, right? That the scales are getting closer to balance there. And I think that, we are starting to see a change in science as well. I hope it happens fast, but it would be great if both of these areas are starting to see equal participation, not just gender-based, but also socioeconomic status, ethnicity, all these sorts of different things. But then the idea that they would work together is really exciting.
It is. But here's the other side of this, though. I mean, is I've seen the lifelong biology enthusiast who's turned into a screen time coder, going, I love what I do, but I'm in front of a screen all day instead of out in nature, which is what got me into this in the first place. And then I've also seen the coders who are like, I'm having to learn all of this biological stuff that makes me uncomfortable. But it's
yeah, well, computers are a huge part of every job now. I mean, I got started because I love animals, and I spend most days, most of the time, in front of a computer. I feel like that's just the way that the world is going, and there's very little to change that.
It's information.
And bioinformatics, I mean, honestly, kids, there are jobs there in the future.
Yeah, absolutely.
So many jobs there. But I like the point of thinking about the number one rule I tell everyone when they come into my lab is that their physical and mental health come before anything else. And that's that is a thing. Like we have to also make sure that we're not just sitting all of the time, that we do get out, that we do interact, that we go outside, that we see the biology that we're working on and hopefully preserve it. And
Mhm.
so,
Absolutely.
yeah, I'm not very good at that myself. So do as I say, not as I do, but I think that's also an important thing in science.
Absolutely. For getting back to coming to the end of our time with you here, and I would love to know what you think as from your research and what you're working on. What are the big questions that are outstanding for you in what you do?
So the biggest question that we have a paper coming out in a couple weeks on is why are there consistent sex differences in certain diseases? So why do women get autoimmune diseases at rates much higher than men? So for example, rheumatoid arthritis, women are diagnosed with RA, rheumatoid arthritis, 8 times more often than men.
Oh, wow.
And nearly all autoimmune diseases show a female bias. On the flip side, nearly all cancers, all non-reproductive cancers, nearly all non-reproductive cancers show a male bias in incidence. That is, females get less cancer than males. And we've been working on a theory to try to explain this, but we're going to have to spend the rest of my career to try to figure out if this is true. Thinking about so if the immune system is so critical for many things, for helping us fight diseases. We know immunotherapies are becoming really popular and beneficial in treating cancers. So if for some reason in women the immune system is prone to being overactive and leading to autoimmune diseases, it could be that it's not that men are getting more cancer, but that women are getting less, because on average their immune systems are slightly overreactive. And so then you would say, well,
And
why?
that explains the autoimmune prevalence also.
So it could, but then we're trying to think about, so what's the evolutionary reason for that, right? Why is there this large difference? And one of the things, so have you all talked about the hygiene hypothesis?
Oh, that's
Yes.
coming up later in the show.
And
But
it's one of the stories in the second half of the
Yeah.
show.
But yeah, bring it.
Okay, can I
Yeah.
flash it? Right. So the
Yeah.
idea is that between for almost all of human history, right, we were exposed to parasites and pathogens, and we didn't have antibiotics, and we didn't wash our hands, and that in industrialized societies we have access to soap and water and clean lives and sanitation, and so that can explain why nearly everyone is more prone in industrialized societies to getting asthma and allergies, right? So the immune system is primed for all of human history, for everybody, to be exposed to parasites and pathogens, but it doesn't explain the sex difference. So the other thing that we think is different is that for nearly all of human history, there was no contraception. You were pregnant from the time you were reproductively active, you were pregnant or lactating until you couldn't anymore. This is our hypothesis. And so the immune system in people who could get pregnant had to be primed, because there's this cool thing. So the placenta itself actually blebs off these little signals. It's trying to downregulate the maternal immune system, because the maternal immune system is saying, hey, right, yeah, like not self, parasites, pathogens, get rid of it. But in XX individuals who have this cool genetic XX mechanism, they have to not just downregulate, but they have to compensate in particular ways, because you have to tolerate this genetically distinct individual trying to downregulate your immune system and also not die of parasites and pathogens. Right? Don't die. Like, that's the goal. Don't die. And now in modern industrialized society, you're not pregnant or lactating for your entire reproductive career. And so it could be that the immune system is primed to be upregulated in certain ways, which is why not all autoimmune diseases show the same sex difference, but it could be primed to be upregulated. But it's not all bad news, because maybe it's leading to those reduced rates of cancer. So if we could explain why there are these big sex differences, and if this is it, then it could help us nail down the mechanism, right? We're not saying everyone should be barefoot and pregnant your whole life. No, we're saying we should figure out what parts of the immune system are the ones that are causing the sex differences. Then we can target those. We can explain why women are responding better to immunotherapies than men. We can explain why they're getting less cancer. We'd explain why men are getting less autoimmune disease, and then we could target therapies. So we just got to solve human health. That's all I want to do.
Just
That
a
is a
little
big-
Human
task.
health and history.
Yeah.
Big outstanding question. It's amazing. All right. Well, I won't keep you any longer. I would love to know where people can find you online if they're interested in finding out more about your research or following your social media and science communication work.
Yeah. So it's sex CHR chromosomes, so sex chromosome lab. Wait, sorry.
I know.
Yeah, lab, sex chromosome lab, CHR lab, either at sex chromosome lab on Twitter or sexchromosomelab.org, those are probably the two easiest places. I do respond to people. I blog. I used to blog all the time before I had that monster that's running around. And that's Math Bio Nerd. Oh yeah, there I am. Look at that. You sound super fast. So yeah, on the main page, it has my Twitter handle and email, and I'm super happy to talk with people. Sometimes I'm very slow responding, but I will.
Fantastic. Thank you so much for joining us tonight. It has been just wonderful to get to talk with you.
Oh, this is awesome pasta. I was gonna hang around, but I'm gonna wrangle some offspring.
Yes.
Some child energy needs to be
some
dealt with.
F1. Yeah.
Thank you once again. Have a wonderful night, and I will be in touch later.
Yeah, thank you very much. Have a great night.
You too. Night. I hope that we can have her back on the show again at some point, because that was an interview that probably could have gone on much longer.
Yeah,
it was just
absolutely.
a fun conversation. I told Blair before the show started, I said, I think she's my new favorite.
Careful, Kiki. Other
Wait.
guests will hear you.
Yeah.
Yes, they hear me. I know. What?
Well, I don't think I remember an interview that was by itself seeming like an introduction to all of the things we could be talking about the entire way through, and it went like an hour. So that's a lot. That's deep.
Yeah, we didn't get to talk about my color blindness. We didn't get to talk about sex-linked traits. Right? We didn't get to talk about temperature-dependent sex determination.
Chromosomal inactivation.
These
Oh
things
my
are all
God.
brought up.
Parthenogenesis. I know, but we really did. There's lots more to discuss.
There is. So maybe we will be able to get her back on the show again another time. If you out there liked what you heard and want to hear again, let us know. And let us know if we should get in touch with Dr. Wilson for another interview sometime down the road to talk more about sex and biology, because this is interesting stuff. We have come to the end of the first half of our show. We're going to take a short break, and we'll be back in the second half with more of that hygiene hypothesis stuff. We're going to be talking about that. We're going to be dropping some coral, and then we got fish skins, because fish skins. Yeah. This is This Week in Science. Stay tuned for more coming right up.
Can't explain things you've heard, more than intuition. A lot of research shows the way to go. New conclusion. The methods of hypothesis and patience are the only things I need. Put on a pair of goggles and go.
Thank you for listening to this episode of TWIS. We would love to invite you to Santa Fe with us. Come to Santa Fe. Are you in Santa Fe? Are you in Albuquerque, New Mexico? Come to the Interplanetary Festival, which is organized by the Santa Fe Institute, taking place the weekend of June 15th and 16th. We will be podcasting live from the Santa Fe Interplanetary Festival on June 15th, 4:15 p.m. to 5:45 p.m. in, I guess that's Central Mountain Time, Central Time, whatever New Mexico time is. We're going to be there. And if you're there, come say hi. We would love to meet you in person. Additionally, if you enjoy what you hear in this show and you are a subscriber, consider supporting what we do so that we are able to do more of it. And we have lots of ways that we make it possible for you to help a little science podcast out. One way is to head to twist.org, which is our website. You can find all sorts of wonderful things at twist.org. Among them, a link to our Zazzle store. At the Zazzle store, you will find all the Twist goodies. We've got bags and t-shirts and pillows and my mouse pads and mugs, wonderful things with the Twist logo and also with artwork from Blair's Animal Corner calendars of past years. Back at twist.org, there is also a link to support us on Patreon. Patreon is our crowdfunding community where you can support us at an amount of your choosing that is charged to whichever account you put into your account at Patreon one time per month. You click on the Become a Patron button. It will give you an option of various levels of support and your choice. $10 a month is a very popular level of support. It's about the same as a cup of coffee or something like that. Less than a soy chai, I think, at most cafes. $10 a month is a nice round amount that brings you into TWIS because we say your name at the end of every episode as a thank you. So $10 a month and above, every level above that, we will say thank you on the air if you help us out there. At twist.org, also, you can click on the subscribe button. So if you haven't subscribed, please consider subscribing. You can click there. It'll take you to YouTube, iTunes, or the Google Play podcast portal to subscribe. If you're looking for another location, you might try searching your favorite browser, your favorite search engine. Also, you can send anyone who is not subscribed to TWIS to twist.org to let them click on that subscribe button and join us weekly. Get TWIS in their inboxes every single week. All the science, more science, more science, more science. And it's all because of you. Yes, all of you help us do what we do, listening, bringing us what has science done for you lately, and all sorts of fun, giving us recommendations for interviews, telling us how it is. And you listen, and this is for you, and it's all about you, really. We couldn't do it without you. Thank you for your support.
Can't explain things you've heard more than intuition. A lot of research shows the way to go.
And we're back with more This Week in Science.
Yes, we are. And it's time now for This Week in What Has Science Done for Me Lately? That's right. Everyone channel channel your inner Janet. Our letter this week comes to us from Joyce Burkers. She writes, "Hi, all. I got turned on to TWIS in my undergrad as a biochemistry student, hearing my professor mention This Week in Virology. I think it was. And that's another very good podcast. And so I went looking for the broader science version. Now, nearly 6 years later, through teaching ESL in Taiwan, 2 years of teachers' college, and moving to England to teach secondary school science, listening to TWIS on long walks or trips is the one constant that has stuck with me. Listening to a recent episode, something clicked in my brain as to what TWIS had done for me this past year. The school I'm teaching in has had a large turnover of science teachers over the past couple of years. So when I arrived in September, most of the science lessons most of my students had received had been delivered by non-science specialists who didn't have the same passion for science as their own subjects. This had led my students being highly unenthusiastic about their science lessons and very closed-minded about science potentially being any fun. Through September and October, getting the students to engage with the lesson was like trying to pull my own teeth out. I can't do this. I'm no good at science anyway. There's no point of trying, were things I would hear every day. I could see why a lot of their teachers had left after short periods of time. It was taking my excitement for science with it. However, I had a half-hour walk to school every morning during which I would listen to TWIS and hear some exciting thing happening in science. I had a bit of backlog to work through, so it was never ending stream for me. This meant that every day I would have some new cool science thing to hold on to and keep that ember of science joy alive in my soul.
Nice.
Just before Christmas, I realized that my toughest group of students were no longer the dread of my day because they were hopping onto the science train,
Yes.
enjoying the experiments and making connections between what we were learning in class and the world around them. In February, I had a student comment, "Miss, your brain is so full of science, isn't it? I bet if your head exploded, all kinds of science stuff would come out." Last month, I had a student tell me, "Boring? Organic chemistry was fun." After being told that a lot of people struggle with it because they find it's less interesting than other topics. And finally, just today, I had a student ask me, "Miss, do you think I could be a person who does science when they grow up?"
Oh, yes. Oh, yes. Oh, yes.
Listening to TWIS has kept my passion for science alive through some really rough days, and it has allowed me to spread it onto a new generation of students who may otherwise have fallen through the cracks and would never have known all the things science does for them every day.
Nice.
Thank you for teaching. Thank you for being a font of scientific knowledge with a head so full of science that it would come exploding out of your head. Oh, thank you so much for writing this letter, Joyce. I really appreciate receiving it. We're so glad to
Amazing.
have you in our audience and listening to TWIS along with us. Thank you. And remember, everyone, if you would like to write in a song, a letter, a note, a sonnet, you can send me an email, Kirsten, Kirsten, at thisweekinscience.com, or leave a message on our Facebook page. That's facebook.com/thisweekinscience. All right, let's get on to that science that we keep talking about. Speaking of the hygiene hypothesis, it's like she knew. Researchers
Oh,
Yeah, researchers just published in the journal Psychopharmacology. And you might be going, psychopharmacology and hygiene hypothesis, what do those things have in common? Why do they go together? Well, so as Dr. Wilson explained in the first half of the show, the hygiene hypothesis is this idea that we're in this modern sterile environment and our immune system is looking for something to react to, right? It wants to keep us safe, but we just don't have anything there. Well, there is a researcher at the University of Colorado Boulder. He and his team have been working on this idea of the hygiene hypothesis and looking at they've done work that we've reported on through the years related to finding that people who live in rural environments, if they work among livestock or around animals, that they seem to have less allergies and asthma than other people, and it backs up this hygiene hypothesis. And the other ideas have also gotten into even further the idea that there are microbes in the soil that are very involved in this allergic or lack of allergic or asthma response. But Dr. Lowry from University of Colorado Boulder, he and his team, they're thinking that maybe it shouldn't be called the hygiene hypothesis anymore, and that maybe we should refer to these microbes in the soil as old friends. And that perhaps we should be changing the idea to it's not that our body is constantly looking for the bad bacteria to fight off, but maybe there are also beneficial bacteria that enable us to be healthier, that through their own actions and interactions with our physiology, they help us be better. And so Lowry had previously published a paper in the Proceedings of the National Academy of Sciences looking at a bacterium called Mycobacterium vaccae. And this
Bakey. Yes.
vaccae,
Bakey. Sure.
okay. And this is a soil bacteria. It's found in the dirt. They found that if they injected this bacterium into mice, they could fend off PTSD-like symptoms and a syndrome after a stressful event. So if prior to the event, they injected the bacterium into the animals and then stressed them out, that the animals were more able to deal with the stressful event and they were less anxious and stressed out and they didn't get the PTSD. And so these researchers are like, whoa, this bacterium is blocking this anxiety fear response. How is it doing that? And this new paper talks about a novel lipid that they have discovered, a fatty acid called 10Z hexadecanoic acid. And it's used, it's inside the cells. It acts like a key in a lock. It binds to specific receptors. They're called peroxisome proliferator activated receptors, PPAR, and these inhibit inflammatory pathways. So they inhibit the body's inflammation response. And so the bacteria, it seems, Lowry says, that these bacteria we co-evolved with have a trick up their sleeve. When taken up by immune cells, they release these lipids that bind to the receptor and shut off the inflammation cascade. And so what he's thinking is that maybe we could develop a stress vaccine from these bacteria that could be given to people in high stress jobs that would help them to not get things like this PTSD type symptoms.
So we would get a stress vaccine from the Mycobacterium vacui, which would make it a double Latin root for cows, and yet cows are not part of the story.
Cows are not part of this story. However, this bacterium is probably found in the soil where cows live.
Oh, right? Yeah, it could be. Yeah.
But the researchers also think that this is just one species of one type of bacterium that is in the soil, but there are all sorts of others, millions of other strains. And in terms of identifying the mechanisms through which they have evolved, we have evolved with them. To be healthy, there are potentially a lot of places that we can be looking for this kind of mental health help.
So okay. So just in as we have talked about, discovering nutrition has this gigantic microbiome element to it. That cognition, health, DNA expression, microbiome plays a role in all of these things. Why should it be surprising that the entire rest of our environment externally is not also contributing, interacting, playing on, and being part of human health for the millions of years that was our external microbiome in effect, right?
I think the idea is not it's not that it's new, it's just becoming more nuanced. So the hygiene hypothesis came about in the 80s, but it's thinking of us responding to the bad environment, right? We're responding to the stressors, we're responding to the bad things that are coming to attack us, as opposed to this more nuanced idea that, yeah, there are parasites, there are bad things out there that are infecting us and doing damage, but there are a lot of good things as well. And so we have to have a much more, I think it's maybe not nuanced, maybe that's the wrong route, but more holistic in our understanding of how the environment influences our entire physiology.
Yeah. So there's an external probiotics that has been overlooked, just as the internal probiotic has been overlooked. And I think essentially at the end of the day, we will find that the majority, I won't say vast majority, because there's not very many majorities that aren't vast, but the majority of microbial interactions over the evolution of life on this planet are positive. That's why we're alive, and that's why they're there, and that's why this interaction has gone on as long as it has. It's the outliers that have the negative affect, and so when you eliminate an internal, if you take antibiotics every day, you will die. Yeah.
Right.
So it shouldn't be too surprising that if you try to sterilize your environment,
Mhm.
you will also encounter negative health effects. This is where we found a healthy balance on being on the planet, is relying not
Yeah.
just like tolerating or being able to handle the exposure to, but actually relying on input from internal external microbes for our health as resources.
Yeah, I think it's an interest. It's just kind of looking at it from a different direction. So, as opposed to saying that, well, some stress is actually good for you, saying it's, if you don't have any bacteria to fight, your body has bad responses. Instead, you're saying, actually, there's good stuff going on that's getting missed out of the picture. So many years ago, we talked about a story from some Scandinavian country. I do not recall which one. I want to say it was Sweden, but they talked about how well, yeah, I guess not Scandinavian, but but anyway, it was a country where they did a study on houses with dishwashers,
Yeah.
and they said that houses with dishwashers had higher rates of allergies. And so the implication there was that by removing the potential allergens in such a perfect kind of hermetically sealed way, they were not exposed and therefore they had higher allergies. But what if, according to this, instead of just being exposed to the allergens, you're actually missing out on an important part of your microbiome that is on those dishes?
Right. And it's possible. Yeah. And so what this guy wants, he prefers calling it the old friends hypothesis as opposed to the
That's
hygiene hypothesis. And
that's
I
all.
think that they may be complementary hypotheses, not necessarily that they cancel each other out. But yeah. So anyway, old friends. We've got our old friends. And then my other cool story had to do again much in line with our interview from earlier. Researchers have been looking at from the Donnelly Center in Toronto have been looking at genes and how they are similar across organisms. And they found that dozens of genes that were thought to have similar roles are in fact unique to humans and might explain our origins, or at least, the origins of all of our amazing abilities like in cognition and whatnot. So these genes, they're transcription factors. They code for these factors that usually are short sequences of RNA that go out and control other genes. So turn other genes up, turn them off, and they're these control factors, transcription factors. These transcription factors, when they're coming to a segment of DNA and they're going to bind to it to turn it on or turn it off or do it, regulate it in the way that they will, they recognize something that's called a motif. It's a little part of the DNA code that's part of a gene, and it's this little identification factor. And these motifs are used as they're like the landing sites. It's got this area of the DNA that's like, you come in here, land, let's do the transcription. Researchers had just kind of looked at these motifs and were like, oh, they're similar. They start with the same things and they kind of end with the same things. They're similar enough, they probably do the same thing. And so these transcription factors, we're going to lump them together and they all do the same thing. Well, this study shows that that's not really how it works. Publishing in Nature Genetics, the researchers have looked at these transcription motifs with a new computational method, and they have been predicting even more accurately how transcription factors bind in different species. And what they found really are that there are subclasses. So you have transcription factors that might do one kind of regulation, but there are these parts of the motifs that they are highly sensitive to, which make them, yeah, that's like it's having a more specialized lock and key situation. So instead of maybe having um, recognizing one particular puzzle piece. It's a different puzzle piece. And so the researchers say even between closely related species, there's a non-negligible portion. That means there's a lot of them of transcription factors that are likely to bind to new sequences. This means they're likely to have novel functions by regulating different genes, which may be important for species differences. And so chimps and humans were 99% identical in our genomes, but there are lots and lots of these transcription factors which vary in what they recognize, which motifs they recognize between the two species. So even though chimps and humans might have the same transcription factors, apparently from the outside, the way that we had been looking at it, now with our finer toothed comb, we're realizing that they're not actually regulating the same genes.
They're not regulating the same, and also the bulk of the genetics that when we say that it's 99%, yeah, but so much of genetic code is just like cell function, like basic cell function, basic like so much of
But
it
so
is
much of it is this regulation. It's what
Yeah,
genes are
yeah.
turned on and which are turned
Yes.
off. And so I think understanding this is like a huge deal.
Yes, absolutely.
Yeah. So anyway, finer tuned control of these motifs in these transcription factors is important for the binding and the function of which genes get turned on, turned off, and how species maybe find our differences.
Mhm.
Tell me a story, Justin.
All right. Let's see. Coral reefs, a thing that we talk about on this show. And if you've been paying attention, you already know that half of the planet's oxygen is produced by the corals. And that coral reefs are in serious jeopardy of decline via climate change, aka climatia. And things may be much worse than we imagined.
Oh no.
New study published in Nature Climate Change finds coral reefs are also under threat from ocean acidification.
Yeah, of course.
But the results suggest some corals and coralline algae being algae that's described here as the glue that is holding coral reefs together, cannot survive the expected acid ocean acidification caused by climate change. This is Quoty Voice. Steve Comau, who is now based in Sorbonne Université, CNRS Laboratoire d'Oceanographie de Ville Français, somewhere in France. The results validate previous research on ocean acidification threats to coral reefs. This is also co-author Professor Malcolm McCulloch from the University of Western Australia. The effects on the calcifying fluid were rapid and persisted for the whole year. Basically, declines in coralline algae, that's the gluey stuff, could lead to the loss of important marine species that use the algae as a nursery. They did find a few of the reef corals that could survive an acidification event. But they also discovered that those were already basically set up to survive. They were already resistant to it. This was not an adaptation event. They found zero adaptation to acidification, which means that the reefs that are even available in an highly acidified ocean are going to be extremely limited and may not be anything close to what we currently consider a reef system.
So there's a couple things here. One is you can take some things from these coral that we've identified that are already set up to be resistant to acidified waters, and you can use CRISPR and isolate those things and put them into other coral species, or you could just use those coral species and try to breed them and adapt them to different temperatures and try to put them into these other reef ecosystems.
Oh, absolutely.
But ultimately, I think the thing about ocean acidification that people get kind of focused on is is kind of just looking at this one thing. But you have ocean acidification happening, and you have ocean warming happening, and you have sea level rise happening, and you have pollution happening, and you have reduction of species happening all at once. And I think that is the thing that we need to keep in mind, is that there's this perfect storm brewing. And if we can reduce any of those factors, there's a better chance of a species making it through the other stressors involved.
Yeah.
Yeah.
Although, to your point of using something like CRISPR or genetic alteration or that sort of scenario, it would then be much easier for us to focus on genetically engineering the next generation of humans to live without oxygen. It would be much. Then we just have one species.
You might have a tough time with just like basic ATP and stuff like that.
Yeah,
It might be
all the
tough
energy
without the
requirements.
oxygen.
We'll just turn on the gills.
You'd really have to start at square one. So that's also oxygen, though. Remember.
Yeah, but
It's just
it's in the
the
ocean.
way.
We'll to the
Yeah.
Arctic
So
and
oxygenation is still important.
we have a couple of steps. It's true.
Yeah, with this though, I think it's very interesting to think, if there's no acclimatization
Yeah.
within 1 year, how many will survive till the second year, and will there ever be acclimatization? Will there be adaptation?
Mhm.
Is it within the ability? I mean, if you don't see it in 1 year, what does that mean? Or if it's basically, is it suggestive that if it's just it's not in this species to survive?
Well,
But it's more likely to happen if
Yeah.
you reduce some of the stress factors, which is kind of what I was talking about before, right?
Yeah.
So if you have everything attacking these reefs all at once, there's a lot less chance for them to adapt and grow and be able to survive. So that's one of the reasons why, for example, marine protected areas are so important, because if you can reduce fishing and human disruption in an area, there's a better chance they can find solutions to ocean acidification or sea level rise or rising sea temperatures.
And so the other thing I would throw out there is if you look back at evolutionary bottlenecks, there is some level of adaptation that we see, but much more often we see niche filling after the fact of a bottleneck, where something expands its range or other things move into a territory that was a niche of resource that was traditionally or previously occupied by some other life form. And that sort of evolutionary niche filling that would take place over millions of years, perhaps, will come long after all humans are gone. Just so people understand, there's a pace and a rate of time frame in which these things take place that is not beneficial.
It hasn't been seen before.
I mean, it's like we've discussed before. The Earth's not going to end.
No.
But it might not be so fun to be here.
There is not to be a mammal.
But you
There's
know
something
why
else to
I'm
enjoy,
glad
whatever
here.
happens.
You know why I'm glad here. I'm glad to be here right now.
Why?
Because it's time for Blair's Animal Corner.
With Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels.
What you got, Blair?
I have a story about some horngry bats.
Hungry.
What do you think horngry means?
That sounds like you get really irritated because you're too.
Well, it's not hangry.
No, there's no
That's what I
anger
heard.
in there.
No, I heard horn.
Mhm. Okay.
So this is a story about Egyptian fruit bats and a very interesting arrangement that males and females have made.
Exactly where I thought this story was going.
Yes, absolutely. This is a story coming from Tel Aviv University looking at the very particular interaction that happens between males and females. They looked at 3 captive bat colonies over the course of a year, and they watched as males would collect food. They would go back into the cave where all the bats were, and then the females would steal food from the males right out of their mouths. But why do the males allow this? Well, it
'Cause
turns out
it's kind of a turn on.
the males are often paid back. How, you ask?
Oh, I can guess. Can I?
Yeah.
May I?
Yeah.
With sex.
You're exactly right.
Oh, okay.
And specifically the kind of sex that leads to offspring. Yes.
Oh.
Yes. So there was a strong relationship between producer, scrounger, feeding interactions, and reproduction. Females bore pups of the males they scrounged food from. So again, 3 bat colonies over the course of the year. And so they kind of watched, they had a bunch of different potential hypotheses. They thought maybe it was relative related, or that there was just so high a cost to defending your food resources, the ladies would beat them up so badly that it wouldn't be worth it. But no, it looks like it's all about sex. So the way they did that is by watching the bats, but also determining paternity that were born in the colony. Females gave birth to young of males from which they had scrounged food pretty consistently. So not only were they having sex, but their babies were from these males that they took food from out of their mouth. There was almost no overlap between males preferred by each female also. So each female had her food bat, was also her boyfriend. And so this looks like the females have specific males that they pick to scrounge from based on individual preference. That next part is the thing we don't know anything about, what the individual preference may be, if it's luck of the draw, if it's just it's easier to go after the same guy all the time, who knows? Especially because those personal preferences change from year to year.
So I'm still hung up on something that happened in the first 2 or 3 sentences of the story, where you pointed out that it was the type of sex that specifically leads to reproduction.
Yeah.
Because I didn't know that bats engaged in the type of sex that could not lead to reproduction.
Well, all animals have
all of them.
tricky stuff going on.
Okay.
It could be sperm storage. There could be sperm competition. There could be just termination of pregnancy. There's so much stuff going on that it doesn't always lead that way. Also, it might be possible females were feeding from multiple males and not all of the males ended up fathering offspring, right? But in this case, they picked one male, they went with him, and that was the father of their babies.
Nice.
I think I find it interesting because there are many species of animals that give food offerings as part of a courtship ritual. So look at what a good provider I am. And so in a sense, this is a female taking that resource and saying, you are a good provider. And we've traded food and now we will have this relationship.
Yes.
It's also amazing because a bat colony is so
Huge.
heavily populated. And it's just when they fly out, it's like tens of thousands of bats, and then they all go back. And that you're finding the one mate in that crowd.
Right.
That's really fast.
Well, you know what they say, bats think all humans look alike. So it's
but still
so
but really
the
I didn't know there was a monogamy thing in bats. The colony is so big, it just seems like they all fly out. How do you not lose track of somebody?
Yeah, it's seasonal monogamy, so it's only for the season. So they have a new mate every single year. But yes, there is a consistency there. The next step kind of this study for this team, they want to see how these relationships evolve, change, and are established over long periods of time. So that's kind of, Kiki, what you were getting to. Is this a demonstration to say, hey, will you go steady with me? Or is this something that the females just try to steal food from a bunch of different males and they find one finally who concedes? Who is the aggressor? How is this being addressed? And does that behavior escalate over time? Is there a variance in how often they bring them food and how often they eat their own food? Is that a decision that the male makes? There's lots of questions.
Is this a male? Is this male mate selection by whether or not they give up the food in the mouth? Like, I don't know.
That's a great question.
No,
Yeah.
I'm waiting.
Hawks,
how
for
hard do you fight?
example.
Yeah.
I love this that hawks, male hawks are way smaller than female hawks, right? And they will bring a kill to a female and kind of drop it nearby and then kind of run
Fly away.
and get back behind and go, do you like it? And then if the female likes it, they're like, can I come? Okay. Can I come a little bit close? Okay. How about if I come a little bit? Okay. So they bring a present. That's very clearly they're bringing the female a present. And they're saying, will you tolerate my presence? So
Do you like my present?
Yeah. Is this that? Or is the male tolerating the female stealing the food?
Yeah.
So there's this huge, lots and lots of questions to be asked. But in the meantime, some very clear food for sex, transactional behavior happening with bats. There you go.
All right.
Yes.
What's up with fish skin?
Oh my goodness. So this is an anecdotal piece of science news. Alert, alert, alert.
Yeah.
Anecdotal. No
mean a-
body of data here.
Nope.
A story.
This is a story. But it leads to a lot of interesting questions. And so I thought it was worth
This is where all the good
discussing.
questions start.
So this is from Michigan State University Veterinary Medical Center. This is a 1-year-old female Rottweiler named Stella. She was caught in a house fire in Lansing, Michigan, while her owners were away.
Oh no.
She escaped the house, but not until she acquired burns across her head, nose, ear, hind end, and sides of her body.
Poor pups.
They were, a lot of them were 2nd and 3rd degree burns. 10% of her body was covered in 2nd and 3rd degree burns. And she had severe smoke inhalation and respiratory problems. She also developed ulcers and scarring in both eyes because of fire exposure.
This better have a happy ending.
She died. No, I'm kidding. No, she's all right. Stella's fine.
And end of story. Scene.
So you could see how this is looking dire for Stella. But spoiler alert, she did survive. So the Michigan State University Veterinary Medical Center jumped in to help her. And the initial threat was the injuries to her trachea and lungs from smoke inhalation and scarring and burning there. So she was put on an IV. She was given pure oxygen up the nose. And once she stabilized, they had to address these 2nd and 3rd degree burns, soft tissue surgery. They could not anesthetize her because of her respiratory injuries. You need to be able to put an animal on a respirator in order to do anesthesia. And so also slowing her heart rate was a concern for a bunch of reasons. So they couldn't put her out. So they couldn't do traditional skin grafts. So they tried something new. They used Icelandic descaled codfish skin.
What?
Yes, and they grafted that to the 2nd and 3rd degree burns on her body. The tissue of this codfish skin has high omega-3 fatty acids, and so they naturally are anti-inflammatory and antibiotic and all this other good stuff. So they could do these skin grafts without heavy sedation. And this is where it gets absolutely insane. The descaled skin is what makes it from other fish grafts, which apparently there are other fish grafts that are done in veterinary medicine, which I didn't even realize, like tilapia. So they'll use scaled tilapia grafts, which during the California wildfires, apparently they used on a bunch of pets. They're effective, but basically it's just an organic covering. It's like an organic Band-Aid while the skin underneath heals. But the descaled grafts, they stimulated the production of cells and ended up becoming functional living tissue. In Stella's case, these grafts, they can change them out, but eventually these grafts were absorbed by her body and new tissue grew into the graft.
It created a scaffold. Yeah.
Yes.
New tissue could grow into. Yeah.
Okay.
Yeah.
So that's
Fish.
Did the dog have scales?
No, it was scaleless.
Okay.
No. So, just to give you an update on where Stella's at, she still has respiratory problems. She'll mostly have to be monitored for the rest of her life, but she is otherwise doing great. She's resting at home. The skin grafts have taken. All of it is sounding good. And so, this is going to be presented at the Society of Veterinary Soft Tissue Surgery Convention in June. So, they're going to discuss the applications of these scaleless fish grafts in the veterinary medical field at a larger scale. And so that's very interesting, but also that I can't help but wonder, how come nobody's talked about humans with this? That seems
Right.
so
Well,
like I'm part fish now. So neat.
yeah, I mean, maybe because maybe we have skin donors, maybe there
Mhm.
are other situations available, and who knows? I mean, maybe we just aren't aware of this scaleless fish skin
Mhm.
in use for
Yeah.
humans.
Well, where do you think
Um,
mermaids come from?
this is something that is currently being first tested in animals
Mhm.
and then will
Mhm.
be
Yeah.
used in humans potentially.
Yeah.
But this is pretty cool.
Yeah, it
Thanks,
seems
fish.
very cool. Yeah, and I'll just give you this last little uplifting note. This is what the head veterinary surgeon said about Stella. She's a living example that the fire within her burns stronger than the fire that injured her.
Oh, I
Oh,
love Stella.
Cute.
that's so
Yeah. She was a
sweet.
fighter, but that skin graft, that fish skin graft really did it.
Yeah.
There you go.
All right, we are at the quick stories part of the show, and I would love to tell you a couple stories about planets. Planets, planets, planets. That's right. So, 18 more exoplanets have been discovered in the Kepler data. This is very exciting. Researchers said, hey, let's look at this in a different way. And so they've discovered in our Milky Way galaxy 18 more exoplanets, one of which is bigger than the Earth, but in a habitable zone where it could potentially have
Mm.
liquid water. It's called EPIC. 202 2012 0.02, but I'll just call it EPIC.
Epic. Okay, did a 12-year-old boy name that planet?
Possibly. It's a
Okay. It
12-year-old
is
boy
possible.
that turned
Yeah.
into a scientist.
Yeah, there you go. Touche!
Yeah, the new technique that they have used, they will be applying to more of the Kepler data. They haven't looked at all the Kepler data yet, so more exoplanets may be in the findings. And then another planet found by a different exoplanet survey called the Next Generation Transit Survey. A this is located at the European Southern Observatory's Paranal Observatory at the Atacama Desert in Chile. And this observatory, it's ground-based. It's not in space like Kepler. It's on the ground. And so ground-based stuff, we know, we're like, hey, what can you actually see? But this ground-based transit survey, looking at planets transiting their stars, has found a Neptune-sized planet in very close to the host star, what should be called, and researchers are calling it, a Neptunian desert. Like, the idea is that you shouldn't find big, big, hot Neptune-sized planets really close into the star, but that's what they found. This planet, NGTS 4b, is 20% smaller than Neptune. It's about only 3 times the size of Earth, and it's been nicknamed the Forbidden Planet because it's not supposed to be where it is. It's hotter than Mercury, 1,000 degrees Celsius, and it orbits its star something like every 1.3 days. So it's just zipping around its star. It's a K-type star. This and the last piece of information about this is that researchers think that this Neptune-ish planet, gassy planet, it still has gas around it, and it's close into the star when it should be irradiated and have lost its atmosphere already, which suggests it either is for some reason really, really strong and able to withstand solar radiation that would irradiate the atmosphere, or it probably migrated from somewhere further out in its solar system to the place where it is close to the star. So Researchers are looking into why exactly it's there, but they're going to look for more planets in the Neptune Desert. Yeah. More stories?
Yeah, I have one more animal story for you.
Awesome.
It's kind of terrifying. Chimpanzees have been spotted in a particular space in Luango National Park in Gabon. They have been spotted cracking open tortoises. They crack the giant tortoises against tree trunks and then just crack them open in half and scoop out the meat. Tortoises and chimpanzees coexist elsewhere, and other primates are known to feed on young soft tortoises, but nobody's ever seen these animals eating full-grown adult hard-shelled tortoises. So this is totally unusual. Interestingly, the same park also has hard-shelled fruit that needs to be whacked against a tree to open it,
Very similar.
which means a lot of the researchers who discovered this think that was actually the kind of start of this behavior, which means this is a particular chimpanzee society that does this. No other chimpanzee groups can do this.
So are these also the chimpanzees that eat crabs? Because there's another story I did. It's not something I brought, but they were talking about chimpanzees that were cracking open and eating crabs.
I think so. Cracking open crabs is way easier than cracking open a tortoise. These are fused bones.
Yeah.
Mhm.
So this is way harder.
Not the same as a
down the
crustacean.
path of smacking stuff open to eat at what's on the inside, this
A
never ends.
crab, you could really just, if you wanted to, you just crack it in either hand. But this is really difficult. And in fact, they even saw weaker chimpanzees passing a tortoise to a stronger adult to whack it open for them. So this is the idea that there is some sort of particular culture of chimpanzees that are doing this, which is a reminder that as chimpanzee populations in Africa are disappearing due to habitat destruction and poaching, that you can't just take chimps from another area, breed them, and put them back in and get the same functional ecological impact as the original society of chimpanzees that was there. Because if they have a particular skill set that they're passing down, that could be lost, which is a good reminder.
Yeah. Justin, you want to talk about Neanders?
Yeah, this is Université France, Anna de Giorne. I'm just guessing. Published in the open access journal PLOS ONE, looked at Neanderthals and sort of following up on the hypothesis that Neanderthals got bred out of existence. And there is also the idea that there was a reduction in sort of fertility that propagated once they started interacting with current modern human out of Africa humans. And what they kind of discovered is that they created a couple of models. Basically, they found that if you reduced the Neanderthal fertility rates amongst Neanderthal women by 2.7%, that wouldn't be enough to make them extinct in 10,000 years, which fits within the 4,000 to 10,000 year frame in which they were sort of here and present in full-on Neanderthal form and indistinguishable beyond some small percentage in a 23andMe genetic survey. If they reduced the fertility by 8%, it hit the low end of 4,000 years. And we have found some evidence within the inheritable traits of modern current humans that fertility related genetic markers that lead to reduced fertility are traceable back to Neanderthal origins. That it's a pretty decent explanation, perhaps, of how they were outcompeted. Yeah, just normal everyday sex.
And that makes us come full circle on our Science of Sex episode. Full closure, full circle. Thank you for listening. Oh my goodness. Thanks for a great show, you guys. That was fun.
Yeah, great show.
That was a lot of fun. I hope everyone out there, I hope you enjoyed it. And I would love to say thank you to Identity 4, who helps record the show, to Gord McLeod, who helps keep the chat room happy, and to Fada, who helps with social media and show notes and also helps out in the YouTube chat room, keeps things above board over there. Thank you for watching on YouTube if you're watching on YouTube, and thank you for listening to this podcast if you're listening as a podcast. I would also love to say thank you to our Patreon sponsors. Thank you to Paul Disney, Richard Onemus, Ed Dyer, Andy Gross, Stu Pollock, Philip Shane, Ken Hayes, Harrison Prather, Charlene Henry, Joshua Fury, Steve DeBell, Alex Wilson, Tony Steele, Craig Landon, Mark Bizarros, Jack, Matthew Litwin, Jason Roberts, Bill K, Bob Calder, Time Jumper 319, Eric Knapp, Richard, Brian Condren, Dave Neighbor, Corinne Benton, Adam LaJoy, Sarah Chavis, Rodney, Tiffany Boyd, John Bertram, Mountain Sloth, Seth O'Gradney, Stephen Albaran, John Ratnaswamy, Dave Friedel, Daryl Myshak, Andrew Swanson, Paul Ronovich, Sue Doster, Dave Wilkinson, Ben Bignell, Richard Porter, Noodles, Kevin Reardon, Christoph Zucknerek, Ashish Pants, Ulysses Adkins, Artyom, Rick Ramus, Paul, John McKee, Jason Olds, Brian Carrington, Christopher Dreyer, Lisa Suzuki, Jim Drapeau, Greg Riley, Sean Lamb, Red Rothig, Steve Leisman, Kurt Larson, Rudy Garcia, Marjorie, Gary S., Robert, Greg Briggs, Brendan Minish, Christopher Rappin, Flying Out, Aaron Luthen, Matt Sutter, Mark Hastenflow, Kevin Parachan, Byron Lee, and E.O. And thank you for your support on Patreon. On next week's show, wait, what else did I want to say? I'm not reading all the things, am I? I'm Distrac-ta-kiki. So, thank you for your support on Patreon. If you are interested in supporting us, you can find information by clicking the Patreon link at TWIS.org. And you can also send your friends who are interested in awesome science podcasts to TWIS.org. On next week's show, we will be back once again. We have an interview with Dr. Matthew Stanely. We're going to be talking about his book, Einstein's War. How relativity triumphed amid the vicious nationalism of World War I. It's got science, politics, war, intrigue, and you'll find out about how the ladies liked Einstein's looks. So many interesting factors. It's got it all. We will be broadcasting live once again at 8 p.m. Pacific time on twist.org/live. You can watch live and join the chat room, but if you can't make it, you can also find our past episodes at our YouTube channel, and you can also watch live on our YouTube channel or at twist.org.
Thank you for enjoying the show. TWIS is also available as a podcast. Just Google This Week in Science in your iTunes directory. Or if you have a mobile type device, you can look for This Week in Science in anything Apple Marketplacey.
For more information on anything you've heard here today, yes, anything you've heard here today, show notes will be available on our website. That's at www.twist.org.
What?
Yeah, it's www.twist.org. While you're there, you can also make comments on any number of things and start conversations with the host, that's us, or other listeners.
Yeah, or you can contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twist.org. Just be sure to put TWIS, TWIS, somewhere in your subject line. Otherwise, your email will be spam filtered. You can also hit us up on the Twitter, where we are at TWIS Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you tonight, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, please remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. This week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be news. That what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma. Got the cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. 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 I've got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you learned anything from the words that we said, then please just remember it's all in your head. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. 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 This Week in 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 Science.
Yes. Distrac-ta-kiki. I'm Distrac-ta-kiki. I need to take more B12 vitamins, maybe. Who knows? It's the after show, everyone. Technically, I think that show was under 2 hours because we started late. I think we actually started the show, 10 minutes late?
Mm-hmm. Yeah, just about.
I think we were about 10 minutes late, and we finished a few minutes around 9:50-ish. So it's a 1 hour, 45-minute long show. Identity 4 will let me know. If that's the case, we did a great job on getting through the stories in the second half of the show.
Yeah.
I'm gonna say, okay. Okay is okay.
I'm gonna share a picture with you. I'm gonna share something with you that you may enjoy.
I like you sharing. Share.
Here, check out this picture of me from 2011.
It's Blair on a horse.
It's one of 3 times I've been on a horse in my whole life.
You're like, what am I doing up here?
It was scary, but also really
That's
fun.
wild animal.
Yeah.
That's Blair on a
pretty
horse. That's awesome.
Yeah. Yep.
You're like happy and scared.
Yep. That is correct. You got it.
Horses are scary. It's true. It's true identity for. I like horses. Some people love horses, though.
They're just kind of scary. They're enormous.
Mm,
And they don't know how much power they have when you're on them.
mhm. And the horses are like big dogs. They're
In
just
some
like,
ways, yeah.
I'm a big animal, and they're just power. They're just powerful. Flying out.
Ugh.
Oh my goodness. Don't punch your horse in the mouth.
Mm.
You don't punch. You just look your gift horse in the mouth.
I've heard you're not supposed to do that.
I've heard it too. Don't do that. Don't do that.
Don't look a gift horse in the mouth.
Don't do that. I have been having some mental time travel issues. I keep removing one of the weeks between the present and when we go to the Interplanetary Festival.
Mm-hmm.
And I keep planning things and I'm like, wait, there's a whole other week in there. Can't. It's good. We're fine. It's fine. Where'd Justin go? He's like, I gotta go. Fantastic in the YouTube chat has brought up the acellular fish skin grafts for management of split thickness donor sites and partial thickness burns, a case series.
Neato.
Fantastic. Apparently is aware of the use of the acellular fish skin grafts for human health, which is kind of awesome. Yeah. Fantastic says Atlantic cod skin has been used for slow healing wounds, which would be kind of like those burns. Right? Third degree, fourth degree burns. Yeah.
Neat.
Yeah.
Cool. I just think it's insane that a completely different species, that skin can be incorporated into
So it's interesting. Yeah, the descaling is one side of it, but when they call it an acellular graft, so what they're doing is they're getting rid of a lot of those the cellular components, but there are the structural scaffold components that remain, and so those allow the body that's injured to have something to grow into and have something that gives its cells structure as the healing occurs. Yeah, it's kind of like in just the field of cellular regeneration when researchers and biological engineers are trying to develop, say a new kidney or a new liver or something. You can't just grow that. You actually have to create a scaffolding for stuff to grow into.
Yeah, I guess they didn't say anywhere in the article that it was acellular.
Yeah, that may not have been. It was without scales. This article that Fantastic had brought into the chat room was acellular.
So they might have just omitted that, but that was really the thing that I thought was interesting. I didn't think it was just scaffold. I thought that it was legitimate fish skin.
Yeah.
Let me see.
I don't know. I'll look at your note. What does it say in the notes? Look in the notes.
Let's see.
They probably give a link to the paper in which they'll give more details about the specifics.
Intact fish skin that is rich in naturally occurring omega-3 polyunsaturated fatty acids. Oh no, this is being used in humans also.
Yeah, okay.
Diabetic wounds. So the fish skin comes from a wild Atlantic cod caught from a sustainable fish stock in Icelandic waters, processed using hydroelectric power.
Wait, what? What are you looking at now?
I'm looking at the website for the brand of the fish skin that they used. It's called Caressis.
Caresses.
Intact fish skin technology. Yeah, I didn't feel like doing a product plug, so
Yeah.
I omitted that. But let's see. When grafted onto damaged human tissue, the material recruits the body's own cells and is ultimately converted into living tissue. Heavy processing is needed to eliminate risk of disease transmission. It is a harsh antiviral treatment, and it removes most of the material's natural components. Yeah, making it dissimilar to human skin.
Interesting.
Mm-hmm.
No need for multi-layer grafts. It is thick. Enhanced cell proliferation and vascularization. Adjustable rate of absorption into surrounding tissue. Wow.
A lot of pluses.
Yeah.
Huh. Hook me up with that fish skin.
Get me from fish skin. How does that work? I want to read more. It's only minimally processed.
Yeah. I don't know.
How does that work?
This story is not listed on their news section yet.
Because it's a brand new story.
Patient stories, it's not there either. It's all humans.
The dog, Stella.
Stella. What a
That's
sweet
so funny.
girl.
I know another Rottweiler named Stella.
Mm.
And our cat named Stella.
Yeah.
Yeah.
Oof. What do you say, folks? Is it bedtime? I'm tired.
Hey, say goodnight, Blair.
Sinking.
Oh, you're sinking. Is there anything that before we say goodnight, is there what,
That would have been bad.
what's happening to you?
Oh no.
Before we go, is there
Oh.
anything you sent the newsletter out, Blair?
No, you didn't tell me I could.
Oh, yeah. I didn't write anything on it. I think I was supposed to do something.
Is it ready to go? Did you approve
I
it?
wrote my thing on it. It's
Oh, okay.
good to go.
I'll send it tomorrow.
Yeah.
I'll write myself a note and I'll send it tomorrow. Okay. You're
I mean,
ready? You've approved of it?
Yeah, if you want to write something else in it, I mean,
Okay.
yeah.
Probably at some point.
But you took out the bios then? Is that
I
what you
took
did?
out the bios.
Uh-huh. Okay.
Yeah. Oh, you're gonna have to okay my
Version.
Oh no, it's letting me in. Oh good.
Yeah, it's only supposed to do that like once a week or something.
Yeah.
Oh, that's very
Okay.
I knew you
TWIS
got in there because I got the
newsletter.
text, but I didn't know.
Good. I did do it.
That's very funny. Will it let us both be in there at the same time? I think it will.
Wait. I hope so. Templates. Wait. Am I in the wrong place?
You need to be in campaigns.
Campaigns. That's why I'm in the wrong place. Okay.
Oh, it might not.
There we go. Number 1.
Yeah, it might not let us be in there at the same time. This might boot you. Let's see.
Give me the boot.
I still don't know where it is. That's okay. Figure it out.
I'm gonna view,
Well, you're supposed to just send me stuff. Just email it to me and I'll plop it in there.
Yeah, you don't have to go enter it in.
You don't have
just
to do
enter
anything
things.
other than just write a thing.
All right, I'll write a thing. Well, I won't make it for this month.
Yeah.
Whatever. But I'll write a thing.
Okay, so let's
Okay,
see.
TWIS.org. Yeah, I've got my thing in there. Da-na-na-na-na. Da-na-na-na-na. Yeah, we could. Yeah,
Oh, this is funny. This
you
is
delete
plenty.
that
I
last.
don't.
It's plenty. Yeah, delete the
Yeah.
last bit about tonight's episode.
Huh?
Okay, let's see.
And
So I'll just edit it to say, don't forget to join us next week on
Wednesday, June 5th
Wednesday,
at 8 p.m. Pacific Daylight Time.
June 5th.
Is there a different one?
Yeah, that's right.
We will
Identity.
be
This is how the sausage is made,
speaking
everyone.
with Dr. Matthews.
I don't want to know how
Uh,
sausage is made. I
don't wanna
like the
know.
fact that I'd have no idea.
I don't want to know either.
Okay,
I'd
great.
rather not.
So should I send it now or should I send it tomorrow? Maybe it's too late to send it now.
Yeah.
Do you want me to just send it now?
Why would you you can send it now?
Okay, because I can just hit send and then it's done.
Newsletter. Hey everyone, you're gonna get a newsletter
Right now.
right now.
Real time.
Ugh!
And if you don't get a newsletter, send me a message and send me an email and I will and let me know that
Okay.
you didn't get a
2.
newsletter. But check your spam folders. Oh,
All
what do you have to do?
subscribers. Personalize the to field. I'm not going to do that. I'm not going to personalize the to field. I don't like that. That's creepy.
What are you talking about?
So you can personalize it to say like to Justin Jackson.
Oh. Why wouldn't you?
I don't know. It's like vaguely spammy.
How about you have it say to my dearest friend?
I'll do first name, last name. That's fine.
Hey, science friend.
Okay, so from This Week in Science, welcome to the TWIS newsletter. This is our first issue of Muslin newsletter, directly from the hosts of This Week in Science. Okay, great.
That's right. Is Washington get rid of daylight savings time?
Send.
Going to mess with
Sending!
everyone. Sending! Yay. Okay, now I'm going
To 219 people.
Woohoo! That's awesome.
Okay, great. So that was our May one. So, but we'll be able to do one after the Planetary Festival. Maybe we'll jot some stuff down when we're hanging out.
Yes.
Together.
Hanging out together.
Hey, how come I didn't get mine?
I just got a
It will go
newsletter.
out shortly. It says.
I just
Am
got
I not
mine.
on the list?
I just got it.
You might not be on the list.
Have you
I
ever
didn't get on
signed
the
up?
list.
Did you ever sign
For
up?
what?
The newsletter?
No.
Well, then you don't get
That
the
would be
newsletter.
why.
How am I but not by default? I don't know where to look at it before it goes out, and I don't get it after it goes out. How am I? You know what? Actually, now that I've recalibrated my thinking, oh well, one less thing to do.
I can put Justin into the.
Thank you.
Put you in
Oh,
there.
yeah.
Mm-hmm.
How silly.
Oh, Blair, I took the TWIS calendar to work.
Uh-huh.
People love it.
Yes.
Not just the pictures, but they love the all the knowing other holidays that they should be
that's great.
celebrating that we should have off.
That's
because it's
great.
fancy. Yeah.
That's awesome.
Add a subscriber.
I didn't get it yet.
I got it.
How weird.
Oh, but my my email program is saying is something. Oh no, there it is.
You got it?
Nope.
No.
But it was saying something was wrong, like it couldn't load other
Um,
stuff that had nothing to do with
Oh, I did get it. Oh, that's fun.
I didn't get it.
Do you want me to forward it to you so you can see it?
At this point.
Yeah, forward it to him
At this
because
point.
we're not going to send it again.
At this point.
There you go.
Hmm.
Our 109-minute long show just appeared in my Dropbox. Thanks, Identity 4. 109 minutes.
Well, because
It's
a
too
bunch of it was
long.
just an awesome interview. I mean, that was and again, I don't recall an interview that was more here's another thing we could talk about. Here's something else we could talk about. Here's something else that would be really interesting to talk about. It was like an hour-long teaser of stories that could be told. Yeah, really
It
exciting.
was. Ha! It's not a scam email, Hot Rod. It's for real. It's for real.
It's
Put it on your.
It's both.
It's not a scam. It's our
Yes, it
TWIS
is.
newsletter.
Yes, it's a scam.
It's not a scam.
It is totally trying to scam you into thinking sciencey things.
No, I wrote a thing.
Mm-hmm.
I wrote a thing.
All right,
Can't
say
wait to
goodnight,
read
Blair.
it. Good night, Blair. Say goodnight,
Is that-
Justin.
wait,
Good night, Justin.
is that all we needed to do? Is that
I don't
is there
Yes.
any other business?
Nothing.
No other business. Nothing we need to discuss.
Good night, Dr.
Good night, Dr. Kiki.
Kiki.
Good night. Good night, everyone. Thanks for joining us for another episode of This Week in Science. We are so excited that we'll be back again next week. I will be doing my Dr. Kiki science conversation on Twitch this Friday at 1 p.m.-ish, 1:15 p.m. Pacific time on my Twitch channel. But next Wednesday, we'll be back doing another episode of TWIS. And we got an interview, and it's going to be fun. So we hope that you'll join us. Have wonderful science. In the meantime, enjoy your newsletter, and we will talk to you soon. Happy sciencing, everyone.